Let's begin. Great. So I will start. Dear all, thank you very much for being here on September round of the transition talks organized by FeMS. My name is Veronika Popova. I'm a plant biologist working in metabolomics, also in transition, but we'll talk about my experience in the next round, which will be in October with other dedicated researchers. For the date, please check our website and social media. Now I will introduce FeMS very, very briefly, in case some of you don't know who we are. Even though we called females in mass spectrometry, we are open for everyone, females, males, non-binary persons, whoever been working in the area of mass spectrometry. We are international community. In the chat, you can find link to our website. We volunteer to support women in the field of mass spectrometry. We do a lot of different things, for example, online events like you are joining today, but also in person and the conferences. On the 1st of September, we had finished registration for the first round of the mentorship. Okay, great. So yeah, on our website, we have a job board. If you want to find a job or if you want to recruit someone, please check our website for the additional information. We also have many awards for different level of people. And I'm happy to say that in the end of this session, we will announce the winner of the Bertha Condron Award. So now I will introduce our first speaker, Giulio Ferrario, who is a PhD student at the University of Milan, studying in the Pharmaceutical Sciences department. Giulio, you can now share your screen and start your talk. Are Giulio here? How it is now? Can you hear me? Hello. Hello.
Hello. Can I start my presentation first?
I can't hear anyone if someone is talking.
Hello.
Great, great. You can hear me. So Okay, so I will again ask Giulio to start his presentation. Giulio, you can share your screen. Oh, okay. So Giulio have problems with his laptop, but I think we can start with Aisha, who is a PhD in analytical chemistry from Nanjing University from China. Aisha, if you have no problem with your computer, you can start your presentation.
Yes, can I share my presentation? In my presentation, I already shared the screen. Hello. Hello. Can you hear me? Vanita?
Yes, I can see your presentation, but I can't hear you.
Okay, now I okay,
Maybe only
now
my
I can
problem.
start. So okay. So today I discussed shortly about the multi-talk and his application for the development of matrices for the analysis of the biomolecule. So I just share one of the relevant one relevant to the multi-talk for the mass spectrometry field. So here, firstly, I discussed about my previous experience. Like I spent I spent 3 years as a postdoc researcher in Suzhou University. In this university, I worked as a postdoc to train the students of the masters and PhD students and working in the biosensor application by using of the search by using the search from spectroscopy. And another 8 months I spent in Yale University in this institute of Changchuan. I spent 8 months and use the ESI mass spectrometry for the application of different food in a light detection. And my PhD from Nandini University in the field of analytical chemistry. During my PhD, I worked in the multi-talk FeMS for the biomolecular detection and the proteomics application. And from Pakistan, I finished my MPhil in the field of analytical chemistry and inorganic chemistry. During this 4 year, I spent uh, to develop different matrices for biomolecules, some of the digestion application, and my MSc is in inorganic chemistry, and bachelor is from University of Karachi. So from the last couple of years, I spent with different journals as a reviewer. from 2016 to 2021. And I have some technical expertise, like during my like more than 10 years, I spent in the research field. So I spent like ESI, HPLC, LCMS, GCMS, and different characterization techniques of XRD and SEM, and some of the multi-top application. So here I just briefly discuss about the multi-top. What is the multi-top FeMS? So here just describe. Okay. Okay, so some basic block diagram I describe about what is a multi-top and how is it deal with the detection of the biomolecule. So here I'll briefly describe about there is the four source ionization, mass sorting, filter, and detection at the sample inlet. So ionization, ion source means the charged molecule. Transfer in the sorting filter, and due to the M by C ratio, they distribute into the analyzer and then ion detected at the ion. So in multi-top FeMS, we use the matrix and the sample and dry on the multi-top plate, and then we use for the MS detection application. Just like in the next slide, just like the multi-top ionization technique nowadays is used for different kind of application because this is a soft ionization technique. But one is the drawback of this technique. First, we have to develop the matrices. So in my previous couple of years, I published like 65 organic matrices used for biomolecule detection and some of the nanomaterial for the protein and peptide detection, and some of the quantum dots for the carbohydrate detection. So first you have first selection of the matrices is very important during the multi-top application because the commercially available matrices is available. But my objective is to try to develop the new matrices, and due to the shorter time, and we have to detect of the different protein and peptides, some of the biomolecule, and some of the sugars in real sample analysis. And in this have the very little fragmentation, and this is a very high throughput techniques. So here I just describe about nowadays mass spectrometry. It's used as a nanomaterial. We have to use for the proteomics based mass spectrometry, and because we have to target the high protein and peptide and increase the abundance of the reduction of the molecule. And nowadays different kind of the MOF, graphene, and metal organic framework, and some of the mesosilica substrate is available for our proteomics mass spectrometry-based analysis. So in my case, I just developed this slide here. I just shortly describe about the different strategies available for this application and to overcome and the problem for the utilization of the matrices and use for the application purpose. So here in my case, I just develop like for proteomics application, we use two different kind of techniques, the multi-top and LCMS. But for both techniques, the multi-top, the drawback is that we have to use the matrices for, but first important is selection of the matrices as compared to the commercially available. But we have to develop the new matrices. But very short time, we have to use to identify the biomolecules by this machine, and we have available different hotspot, available software. So we have to analyze which protein and peptide we have to analyze from the digestive sample, and some of the MS-based techniques is also available, but is takes more time. So here I just use the mesoporous silica to functionalize with sulfonic acid, and is very so by using of the hydrophilic and hydrophobic interaction, So here, just like by using of the FeMS, we have to use a different expensive column separation techniques is available. But here in my strategy, I use the substrate like the mesoporosilica by functionalized with sulfonic acid group. So by manually in the room temperature, I have to set the incubation time period, like the which time period is suitable for absorption of the peptide by mesoporosilica material, and then I have to centrifuge the desired binding the lactopeptide, and then I use to optimize the washing method to remove the unwanted glycopeptide, and by elution method, I deduct some of the peptide available in my sample. Just like in this figure, I just described, like I use the HRP in very low concentration, triple digest of the HRP, and with enrichment of the sulfonic acid group, I detect at least 21 of the glycopeptide by using of the mesoporosilica modified with sulfonic acid group. So here is also I use the picogram molar in chicken evident triple digest without enrichment. Just like I haven't used the mesoporosilica and with the triple digest, and after binding with triple by the mesoporosilica, you have to see that I have to detect by 16 glycopeptide by using of this material. And the time period is at least 2 to 3 minutes. Is all glycopeptide bind with the sulfonic acid group. So here is another example, like if we have to use some mixture analysis, either this material is suitable for the lower concentration. So here the proof of the different ratio, the enrichment time period is also very shorter and very lower concentration. This mesoporosilica is bind many of the peptide by using of this mesoporosilica. So here I just describe about the summary and advantage of the glycopeptide detection by using of the mesoporosilica. So in the initial strategy, I have described about, like I have to optimize the time of incubation and the material functionalization. So here the summarize of the basic points of regarding this strategy. So here, during the purpose of this transition talk, just I describe of the one of my paperwork because due to the shorter of time. So right now I am in transition phase, so I'm looking for different position, like as a research assistant or as a research scientist, to utilize my expertise to do some more work in the mass spectrometry field. And currently, I'm looking for different countries, not specific, but here I mentioned about some specific country where I'm looking for the position, because according to my knowledge, my expertise and research laboratory is available in such countries. So right now, I try to join if I have some opportunity to join as soon as possible to utilize my expertise. And this work is delegated to my professor, Giulio Ferrario. He is the director of the State Laboratory of Energy during my PhDs. He is involved different kind of research project, and still now we have some collaboration work with Giulio Ferrario to help with his master's and PhD students during when I stay at home. Thank you.
Thanks a lot, Aisha. If anyone have a question, you're welcome to ask. If no one have a question, I have. So Aisha, Could you please say what is the most interesting thing you have found for yourself through your research?
After my PhD, just like I think maybe I have a capable to train to other students in the field of the mass spectrometry because during my postdoc and my PhD time period, I learned from this experience from to help or the projects development of the master's and PhD students or writing the projects and also design some different projects for different funding. So I try to explore and utilize these expertise into my new positions.
Great. Thanks a lot. Thanks a lot for your presentation. And now we will, I think we will go to our next speaker, Kelly Conger. She's a 6-year PhD candidate at the University of Illinois in Chicago. Kelly, please, you can start sharing your screen. Aisha, you can close. Great.
All right. Hi, everybody. Let me get my laser pointer going. All right. So hi, I'm so excited to be here talking to FeMS. Let me know miss that. So I actually found FeMS on Twitter, and I've been following this group for a few years now, or a year now. And so I'm really excited to be giving this transition talk. Like she said, my name is Kelly Conger, and the title of my talk today is just Cancer Metabolism and My Mass Spec Future in Chicago. I'm currently a graduate student at UIC in Dr. John Koloff's lab. And kind of the purpose talking today is that I'm looking for biotech and industry positions for early 2024. So about me, I'm from the Midwest in America. I'm from Michigan, and I came to Chicago and completed a bachelor's in science at DePaul University back in 2017. And I really started my research early on doing some Drosophila genetics work. I just wanted to be in the lab and start getting my hands dirty. And it was ultimately not where I ended up in genetics, but it was a great start. And then I stuck around in Chicago, and I'm currently wrapping up my PhD in physiology and biophysics, intending to graduate here pretty soon. And I'm currently seeking scientist positions in biotech, with the caveat that the perfect postdoc position could sway me. And I'll talk a little bit about that at the very end. When I'm not in lab, Chicago is just an absolutely incredible city. If you're familiar with it, I'm usually out and about. In the summer, there's street festivals. There's a million restaurants here. The lakefront is gorgeous. I'm a huge advocate just for the city in general. So that's kind of my real basics about me. I am a little bit different in terms of mass spec from sort of how people typically think about it. So I use mass spec to study amino acid metabolism. Our lab here at UIC is a new basic science lab. So I'm actually my PI's first graduate student. We're a small lab, but we've been growing pretty rapidly. And our general focus over the last few years has been on metabolic differences between tumor subtypes, and specifically for my project, looking at differences between ER positive and triple negative breast cancer. So we're really just using mass spec as a tool to study these more basic science questions. And our first paper came out last year in Cell Reports, highlighting these differences in amino acid metabolism between the breast cancer subtypes. So it's a great little read. Kind of my claim to fame in the lab and what I've spent a lot of my time working on is developing assays to measure amino acid uptake into cancer cells. So I do this using isotopically labeled amino acids. This is not, deuterated or radioactive. This is going to be carbon-13 and heavy nitrogen-15 labeled amino acids. I'm working with a Agilent 7890B GC MSD system. So this is all gas chromatography work. This is not liquid chromatography. And as I said, we're a pretty small lab, so I have been the lead maintenance worker on our mass spec over the last 5-6 years. So I know this thing inside and out. I call it my baby. This is my mass spec right now. The highlight of my project has been identifying, kind of for the first time, serine transporters. So one of the 20 amino acids, serine transporters in cancer cells. And if you are at all familiar with amino acid metabolism, we've identified ASCT2, or the gene is SLC1A5, as the primary serine transporter. A little bit more about the actual work and how I've been using mass spec. So again, I'm using GCMS to track the accumulation of labeled analytes in cells. And I've just thrown up this little figure here. So we're able to use CRISPR knockout cell lines and then track the accumulation of isotopically labeled serine into these cells over time. I've also done depletion assays from cell culture media, as well as collaborating with some friends down in Florida to use LCMS in order to measure purine nucleotide biosynthesis, again, in our amino acid transporter knockout cell lines. And so the real goal here is, how do available nutrients affect cellular metabolism, affect cancer metabolism, and cancer growth? And then ultimately, can we target amino acid transporters for cancer therapy? And so this was just one of our most recent in vivo mouse experiments that I'm showing, where we placed the mice on a serine-free diet in combination with knocking out one of our amino acid transporters. And so really thinking about how that low serine diet affects tumor growth, and we were actually able to achieve regression of almost all of our tumors on the combination treatment. So this was really exciting for us. And we will be submitting, hopefully, my final manuscript for publication next week. So hopefully keep an eye out for that soon. Of the people and places that I've been able to meet during my PhD time, I have a lot of people to thank and collaborators that I've been lucky enough to work with. So Peter Sorger's lab at Harvard Medical has been incredible. His postdoc, Chris Chidley, has a paper on bioRxiv right now where he's using a transporter-specific CRISPR screen. This is really what got my project off the ground a few years ago. Gina DeNicola's lab at Moffitt Cancer Center in Florida. I talked about how we were doing purine nucleotide work. She does a lot of that and more. Ryan Sheldon from the Van Andel Institute. This is a very heavy metabolism-based research institute, and he's really the guy that taught me how to do mass hunter analysis for our GCMS data. And then finally, Issam Ben Sar's lab at Northwestern here in Chicago, kind of keyed us into some ideas revolving around transporter substrate dynamics, which ended up being a whole extra chapter of our work. Some of the places that I've been fortunate enough to present my work. So obviously here with FeMS, I'm also a member of the Association for Women in Science Chicago chapter, and I've been sort of in their mentorship circles this summer. CABTRAC is the Cancer Biology Training Consortium. I'm going to be presenting a poster there in California next month. ASPIT has been a really great new find for me this year. I was selected to present a guppy talk for their 2023 annual conference in St. Louis, where I pitched my transporters as a therapeutic target, as well as doing posters for their Great Lakes chapter just a few months ago, which was great to just see kind of the local support from ASPIT. I presented at Chicago Mass Spec Day, where I kind of went in depth about how we're using GCMS for metabolism. And then finally, the Keystone Tumor Metabolism Symposium, which was perpetually postponed due to COVID, but we finally got to go last September, and it was a really great time. So what's next for me? I will be defending my thesis in November. So the rest of my day is just thesis writing now. I'm looking to transition into a scientist role at a biotech company. However, if there was a postdoc position that was very technique specific, that could kind of bolster more of my proteomics, mass spec, omics techniques, I would definitely go for it. My preferred location is Chicago, but again, perfect position means I'm willing to move somewhere. I'm not only interested in cancer biology, although that is where I have the most background, most experience right now, but really anything revolving around therapy development and testing. using multi-omics, sort of combining metabolism and proteomics to get a really well-rounded picture of what's happening in disease. It was kind of fortunate that Aisha was talking about MALDI-TOP in our first transition talk. I'm an absolute fangirl for MALDI work. I think using spatial metabolomics can be really interesting, particularly when you're thinking about changes, metabolic and proteomic changes in primary versus metastatic cancer disease. That's kind of where my head is at for potentially a next project. I think my strengths in the lab and coming out of the PhD are really in project and team management. I've trained all of our incoming students, our postdocs. I maintain the mass spec. It's a real point of pride for me. And I just really love kind of being part of a small team. Mentorship is obviously hugely important to me. I will be joining the FeMS mentorship pods this summer or this fall. I'm really excited about that. I love both receiving mentorship as well as being a mentor to younger and newer trainees in my lab and in my program in general. And so those are sort of some things that I'm really looking forward to and looking for in a next position. So with that, I think I was kind of quick, but I want to thank the entire FeMS team for putting this on. Here is where to reach me and my little LinkedIn picture. I'm happy to talk to anybody about metabolomics and mass spectrometry in general. So that's my chat.
Thanks a lot, Kelly, for your talk. We have a question in our chat. Aisha asks, do you ever done any CI with your GCMS?
What is CI?
Chemical ionization.
Chemical ionization? I personally have not.
Okay.
But thank you for the question.
So I also have a little question. If you could investigate whenever you want, what it will be?
Yeah, I think I would do, I mean, I'm thinking about this a lot now. You know, my PI is like, where do we take your project after you leave? And what am I looking for? I would really like to kind of interrogate the proteomic and metabolomic changes that occur in brain metastases. So the brain is a really nutrient deprived and unique nutrient environment. And so the fact that we get brain metastases from a lot of different types of primary tumors, I think is really interesting. They become extremely hard to target. And so looking at potential therapeutic vulnerabilities in, I think, brain metastases would be really interesting. Yeah.
Wow, great. If anyone have a question, can ask.
I have a little question. Sorry, my camera just collapsed, so I will try to talk like this. Hi, Kelly. So as far as I understood, so you are focused so technically looking at GC, right? This is
Yes, my experience has been with GC.
Yeah, this is your experience. As you mentioned, your baby, yeah, right? So just my question, in this transition period, it's pretty important to be also flexible. So are you flexible to switch, for example, for LCMS? And you also mentioned proteomics, because this is where you are not working with GC. This is another technology and pretty well developed and advanced. How you see it?
No, I'm very interested in learning LCMS. It's a real area where I feel like I've missed out. I kind of hit the ground running with GCMS, and I see all this beautiful data published with LCMS. I definitely want to get my hands wet with that. I'm looking for any opportunities to learn, and I feel like it will be hopefully a little bit easier with a bit of a GCMS background, a little, you know, kind of conceptual understanding of it. But maybe I'm naive. I am just very excited to, you know, learn as many techniques as possible. LCMS, I'm absolutely open to it.
Okay, thank you. I see your great enthusiasm. Yeah.
Oh, thanks. Yeah.
So now I think we can move to our next speaker, Giulio.
Yes.
Yes, you can start your presentation now.
Okay. Everyone can see it?
Yes.
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 Okay. Okay. Perfect. Thank you so much for having me here. It's a great pleasure to be here. And just from the start, I'm going to in a little bit of help for my transition. I'm from Italy, and specifically I'm from Saronno. I'm sure you're aware about the Di Saronno and the Amaretti, but right now I'm in Piacenza, where normally I have fun in climbing, hiking, and every kind of sport. But during these last years, I've been a PhD at the Department of Pharmaceutical Sciences here in Milan. My career started during the 2019 when I did my thesis. I did an external thesis, so an industrial thesis at Nerviano Medical Science. During this period, I really fall in love with research, and so I wanted to know a bit more about mass spectrometry and about proteomics. So after this thesis period of 1 year and after COVID, I went on and joined my supervisor, which is Professor Giancarlo Aldini, for a PhD. And then during my PhD, I went on and I did a 7-month period at the Karolinska under Professor Per Nordlund for my project, which is mass spectrometric strategies to identify Hello, hi. Sorry, today's my laptop is not behaving well. Should I restart and try again? Maybe from where I stopped. I hope I will not be interrupted again by my laptop. Okay. Okay, so I was talking about my research, which is a bit intricate. I won't be long, but we used some different mass spectrometry techniques. For example, we started from analyzing natural extracts by liquid chromatography. So we did some metabolomics of different components of these extracts. And once the components were known, in parallel, we did some testing with some in vitro assays to understand if these extracts were active as anti-inflammatory agents, and then we used some proteomic to understand like the impact on the proteome of these extracts. And of course, I'm a bit of a handyman in the lab when needed. So the various techniques are very various. So the first approach was done on thin apples, which I don't know if you know, but during for your freshly juicy apples that you find in the supermarkets, there is a specific process which is called the pruning to obtain those apples. But this pruning process generates a lot of waste each year. But this waste is really enriched in polyphenols, even up to 10-fold more than normal apples. So we did an extraction of these apples and we try and test it out. And we found out that had some particular anti-inflammatory activity and also some antioxidant activity vehiculated by the activation of NRF2. And analyzing the impact on the proteome of this extract, we confirmed this activation of NRF2, and also we found out the activation also of the pentose phosphate pathway, so a whole reprogramming of the metabolism by this extract. And during this first work, I learned a lot about sample preparation for mass spectrometry and also cell handling, because I had to do twice the same experiment using Silac, because it was my first time. And then we went on and we said to ourselves, well, if this extract is now active in an in vitro model, could worldwide? But we tried and find out, and we found out that this extract was really active and also had quite an action, an anti-inflammatory and also an antioxidative action in this animal model. So with this experience, I handled different tissue samples that were colon samples. And also it was really tough to make sense of everything, that all the results that we obtained was really hard to connect every dot and get a full picture. But one thing that I also did during my PhD was to apply the TPP or CHETSA, if whatever you want to call it, so target protein profile, for investigating which type of target some of these extracts have, because normally we have these extracts which have a lot of moieties inside, but we don't know which is the real target or the main target in the cell. So we tried to use this approach, and we were quite happy with it, because we had some solid targets, and these targets well translated from an approach where we used a cell lysate, but was really well translated in vivo and using in-cell CETSA. During this period, I understood how to deal with another different type of sample preparation for proteomics, which is quite tricky when you have different temperatures, different conditions. You need to be really prepared. And also use a bit of R, which is always useful. And also to put my hands on Q-Exactive instruments. I always had some experience with an LTQ XL and a Velos Pro. Then during these years, what really I found out that I really liked was to learn a lot, learn new things. And so I tried to go to and attend to a lot of courses and to get a lot of knowledge from my colleagues and everyone in the lab, and also from every single technician that was coming here whenever the instrument was failing or the HPLC wasn't working, because I really like that part of my job. I really like to apply myself and try to find what it's not working and try to do it myself if possible, obviously. Whenever I'm trying to describe myself, I always think about the English saying about if you are a jack of all trades, then you are. But it's the position where I find myself in, because I had a lot of experience with a lot of approaches using mass spec, but I do not feel that I'm like super expert in one part. And as of now, I'm like trying to transition from academia to industry, but lately I've had one work proposal in academia. So what maybe you can help me to understand is like how can I translate my experience with this, if you can pass me the term, old instruments to newer and cooler instruments that you can use, and also if you have some resources that are useful to catch up with these new instruments, because sometimes it's just it's only some stuff, and that's why I'm here to ask a little bit of help if you can. And for me, that's it. And thank you for your kind attention.
Thank you, Giulio, for your talk. If anyone have a question or an answer for Giulio's question, go ahead. From my side, I want to ask you, Giulio, a question about what kind of instruments in what kind of instruments you are dreaming to work with.
I mean, like from the top of my head would be, I think, a team stuff right now. But what I really like is to apply these new instruments to answer some like biological question or something like that. I would really like to go to work myself into like chemoproteomics. So even that type of thing for me is something that I want to be in. But I think any instruments for me is always good.
I can give you one advice. I mean, your chances of getting a job they don't depend on what instrument you are using at all.
Okay.
It's irrelevant.
Thanks a lot. Thanks a lot, Giulio, and thanks a lot for all our speakers today. I wish you many lucks with your job search. And now I would like to introduce you David from the Agilent, who will announce the winner of the Bertha Condron Award. David.
Yeah, thank you. Wonderful to be here. This award is, even though I work for Agilent, is not linked to Agilent. It's a personal award sponsored by myself. Bertha Condron was my grandmother, born in 1911, and this particular award is around a recognition of people more at the technician level. My grandmother, particularly in that era, had to start work at the age of 12. I still have, as part of her paperwork, her release from her parents, where at the age of 12 she went to work in cotton mills where she had to live and work for 6 days a week. They were locked in their dormitories at night, and this was pretty typical of that era. So this particular award recognises that not everyone has full opportunities for education. Not everyone has full opportunities to fully develop themselves in terms of their capabilities. And it honours the life story of my grandmother, who eventually moved on from starting work at the age of 12 to become a mother, a grandmother, great-grandmother, and lead a fulfilling life through other means without education. So this particular award is in her honour, and I'll pass back to you to announce the winner, please.
Mhm. Sorry, could you please repeat your last sentence because I just messed up with the.
Yeah, I'm just passing back to you now.
Oh,
So
okay.
that you can the winner on behalf of FeMS. Thank you.
Oh, okay. So I'm happy to announce that winner is Andrea Schuchmann. Who yes, who, if she wants, can give a talk about her work, please.
Dear audience, can you hear me? Okay. Dear audience, dear FeMS in Mass Spectrometry Initiative, thank you for introducing me. I feel highly honored to get the Bertha Condron Award in 2023. Feels awesome. I would like to thank my group leader, Andrei Shevchenko, and my lab members at the Max Planck Institute of Molecular Cell Biology and Genetics in Dresden for the nomination and the support. The award is appreciation of my work as a lab technician in the field of mass spectrometry since 15 years. In these years, I had the opportunity to work on various projects as a part of a strong team. I enjoyed assisting great scientists in their projects from the start, over the method development to the final result. Over the years, many biological and sometimes also archaeological questions have been solved as a team. And every big and small success was celebrated together. Keeping up with the tradition, we will also celebrate the Bertha Condron Award, and I will take you out for dinner, the whole lab. Furthermore, I would like to take this opportunity to encourage women in the mass spectrometry field, especially master students or PhD students, to take advantage of the various possibilities that females in mass spectrometry offers, like mentoring programs, talks, awards, and the active and strong worldwide network. This support is a big help to progress your future career in mass spectrometry. Thank you all for your attention.
Yes, sound of applause to Andrea. Thanks a lot for your work. So now we just in time. So thank you all for being here today. Now we will stop our session. Thanks a lot again.